Good day, and welcome to DNA Genotek and Diversigen's webinar on overcoming challenges in skin microbiome research from sampling to analysis. My name is Heloise Breton, and I will be your moderator for today. Before we start, we wanted to give you a brief overview of today's talk. So we will start by reviewing DNA Genotek's newest product, OMNIgene SKIN, for the sampling of skin microbiome. We'll quickly look at how this device can be used to profile various skin sites. Then we'll move on and present to you some of the validation data for this product, which will demonstrate sample stability. We'll also look at how you can optimize your sample processing workflows to obtain better results, and also look at some of the bioburden testing and quantitative performance data for this product. Then we'll move on to Diversigen's V4 and Viv3 analytical skin pipelines, and then open it up for a Q&A. But first, let me introduce our speakers for today. Our first speaker will be Brice LeFrancois. Brice is the microbiome team lead scientist at DNA Genotek. He has extensive expertise in molecular biology, microbiome sample collection and processing, and DNA and RNA extractions. For the past 2 years, he's been leading multiple research projects at DNA Genotek, including the development of the OMNIgene SKIN device he'll be discussing today. Prior to joining DNA Genotek, Brice obtained a Ph.D. in biochemistry from the University of Ottawa and has more than 8 years of experience in academic research across a wide range of fields, including cancer and neurobiology. Our second speaker today is Daryl Gohl. Daryl is the senior scientific advisor at Diversigen and was one of the co-founders of CoreBiome, which is now under the Diversigen banner. Dr. Gohl is also the head of the University of Minnesota Genomics Center's Innovation Lab, as well as a research assistant professor in the Department of Genetics, Cell Biology, and Development. Daryl holds a Ph.D. in molecular biology from Princeton University. His years of research have contributed to the development and improvement of amplicon-based microbiome profiling methods, as well as a novel shallow shotgun sequencing approach. Now, without further ado, I'll hand it over to our speakers.
Thank you, Eloise, for the kind introduction. And my part of the talk today is going to be focused on addressing the challenges in human skin microbiome collection and processing. So when you're looking at the human skin, you're looking at a wide diversity of collection sites, and each of these sites tend to support different microbial community. And typically you can subdivide the human skin into 3 main groups. So you've got sebaceous skin or oily skin, and that's typically the face, scalp, or the chest. Then there's also wet or moist skin, which would be like toe webs or any body folds really. And then there's dry skin, so that would be hands, arm, and legs. In general, skin is very low in nutrients, and as such, it has very low relative microbial abundance. So for human skin, we're typically talking of somewhere between 10 to the 3 to 10 to the 9 bacterial cells per square centimeter of skin. And dry skin, which is the lowest biomass skin site, tends to be on the lower end of that range, while sebaceous skin is closer to like 10 to the 9. And just keep in mind that it's several order of magnitude below what you typically encounter on other human microbiome sites, such as the oral cavity or the gut. And since skin is such low biomass, environmental contaminant and bioburden, so bioburden is background DNA contamination that would be coming either from the collection device itself or the extraction kit, can actually be significant issues in your analysis. And to this day, there's a lack of a standardized means to collect skin microbiome sample. And there's 2 main methods that are being used by researchers out there. So one of them is swabbing, where you're using a swab. And some people argue that swabbing is not as good because you're only going to be collecting the surface microbiome. And the second approach is to use tape stripping, so using a sticky tape to capture the bacteria that are present on the human skin, and that allows you to do deeper sampling. But extraction can be a challenge from tape stripping because it's very hard to dislodge the captured microbial cells from the sticky tape. So we decided to leverage all the internal knowledge that exists at DNA Genotek on microbiome collection devices. And we developed OMNIgene SKIN, which is a swab-based skin microbiome collection device And it's a very simple device. So as you can see, it consists of a flock swab with a breakpoint, an OMNIgene collection tube, so that contains 1 ml of stabilization buffer, and also a wetting solution that's going to be used to moisten the swab prior to collection. And included in each kit too is like our optimized instruction for use. And they don't look like much, but we spent a lot of time developing those IFUs because we wanted to make sure that the end user collecting the sample at home was going to do as good of a job as possible. And as you can see, the steps are very simple. You take the collection tube out of its packaging, same for the swab, then dunk the swab into the wetting solution before collecting the sample from the site of interest. And I think that's a key step where we're asking the end user to collect for a total of 60 seconds while applying a lot of pressure. And applying that pressure and the increased collection time is really making sure that you're going to be capturing as many of the microbial cell on the skin surface as possible. And it's going to ensure good performance in downstream analysis. Very early on in the development of OMNIgene SKIN, we realized that it was paramount that we would develop workflows that would be able to overcome low biomass. And we needed to make sure that we had appropriate workflows so that we would get enough DNA from any skin site that we want to sample. And we ended up developing 2 workflows that are shown here in this figure, where you start with an OMNIgene SKIN sample and you do a proteinase K treatment for 1 hour at 50 degrees, where you've got the swab sitting in a stabilization solution. That step is going to help releasing bacteria into the stabilization liquid so that it's available for downstream extraction. And when it comes to extraction, there's 2 ways of processing those samples. The one that we recommend is using PowerFecal Pro, which is a QIAGEN kit that works very well for most microbiome sample. And we suggest that people concentrate the 1 ml of sample down to 250 so that you can process the entire sample in one step. But we also developed our own extraction protocol, so DNA Genotek in-house protocol, where you can skip that concentration step and take the entire 1 ml stabilized sample straight into a bead beading tube, and then you can isolate the DNA using silica columns. And as you can see on the graph here, showing you typical yields we get from several skin sites using both workflows, we were able to achieve significant DNA yields from each of the skin sites that we sample. So for sebaceous skin, whether it's scalp or the face, you see that on average we recover somewhere between 100 to 200 nanogram per kit, which is pretty good. For forearm, which is dry skin, so very, very low biomass, you see that the yields are much lower. We're talking about 10 to 20 nanogram per kits, but it's still pretty good when it comes for dry skin. And then wet, moist skin sites, you see that it's very donor and site dependent. So we did see pretty high yields for some donor for toe webs, but in general, axilla or the armpit were much lower. And included in any of our experiments are environmental control. So environmental controls are uncollected kits that go through the extraction. And as you can see, our environmental controls gave very, very low DNA yield, which indicates that the bioburden in our collection device is pretty low. And it's also important to point out that the yields that we typically got from collected sample from any site were 95% of the time higher than those environmental control, which really indicates that we're collecting the material that's present on the skin surface of the donor. The next thing that we did was check the performance of the extracted DNA in amplicon sequencing applications, so both 16S and ITS. And in this graph, I'm showing you final library quants for samples that were amplified with 16S ITS for face, toe web, and forearm. And as you can see, we got pretty robust amplification and final library quants for both 16S and ITS for toe web and face. We saw slightly less amplification for 16S for forearm samples, but that's expected because yields are significantly lower. But importantly, again, when you do the same PCR amplification on our environmental control, you see that the final yields are very, very low. And that's again showing that the bioburden in our kits are very, very low. We barely detect any amplification with 16S. So anything that you're generating in those libraries corresponds to materials that was extracted from the skin of your donor. So the next thing that we wanted to do was to check that OMNIgene SKIN can capture the site-specific bacterial and fungal profile. And here I'm just showing you bar plots showing the relative abundance of the genera that we detected for like several skin sites. So all of our data was generated using 16S taxonomic profile using V3, V4. And as you can see for face and scalp, for most donors, it's dominated by Cutibacterium, or also known as Propionibacterium. While when you look at the toe web, you see that it's a different story, and there's two genera that are dominating the samples. So you've got Staphylococcus in gray and Corynebacterium here in beige. And Corynebacterium is expected to be seen on moist skin site because it's a moist-loving bacteria. And in general, if you look at the profile that we generated from OMNIgene SKIN samples, they're very much in line with what's been reported in the literature in the past. So you see, for example, that when you do sampling of sebaceous skin site, it's mostly dominated by Propionibacterium, whereas when you look at toe web or dry skin site, it's a different story. And you see, for example, Corynebacterium and staph in the toe web, just like we did. So that's really good because we're capturing the specificity of each of these sites. And this is like the same data, but showing you the ITS2 taxonomic profile and the relative abundance of fungal genera. And you see that forearm, face, and scalp samples tend to be dominated by Malthasia. So it's a skin commensal, a genus that's like very well established to reside on the skin of humans. And as you can see, it's like the most abundant fungal species across most sample sites. But again, the toe web samples were quite different, and we were able to see like a greater diversity of fungal signatures and genera in them. And that's again something that's been reported in the literature. So OMNIgene SKIN really can capture like site-specific fungal and bacterial profile. With like our optimized processing workflow, we were able to achieve like pretty high yields for like select skin sites, so mostly face, scalp, and like toe web. So we decided to do whole genome sequencing on a subset of those samples. And I'm showing you in that graph the taxonomic profile and the relative abundance. And as you can see, the data is quite similar to what we saw with like 16S amplification. And you see that face and scalp samples, which are like sebaceous, are dominated by Propionibacterium or Cutibacterium again. But you see that in general, the proportion of Cutibacterium is higher than what we saw with 16S. And it is likely because we don't have any PCR bias when we're doing WGS sequencing. And when you look at the toe web samples, again, you see that it's mostly dominated by two species. And those two species are going to be Staphylococcus and Corynebacterium in green, as expected. And if you start to look at the clustering by PCA plot, you can start seeing that each of the sites is going to cluster, and each site is clustering with itself. So you see that the toe web samples tend to cluster on the right hand of the plot, whereas face and scalp, which are highly similar, tend to be found on the left side. But you're still able to like start seeing like differences between the scalp and the face, which is really exciting. And that's what Shotgun gives you, like better resolution into what's present on those different sites. And since Propionibacterium is like very well known to play a role in acne, we decided to look in more details into our Shotgun data, and we were able to identify up to 20 unique C. acnes strains in those samples, which is like very exciting. And we decided then to like organize those 20 unique strains based on the ribotype. So ribotype is shared 16S sequences, and strains that are very closely related tend to share their 16S sequences. And as you can see, when you look at our 10 donors, they're mostly dominated by ribotypes 1, 2, and 3, which is expected in healthy donors, but we were also able to detect ribotype 4 and 5, which are known to be enriched in acne patients. And here they're low because we're looking at skin samples from healthy individuals. So over the next few slides, I'll take you through some of the data that we generated during the validation of OMNIgene SKIN. And as I mentioned in the introduction, like skin has very low microbial loads. So you cannot sample the same site twice because you're going to end up depleting microbial cells with the first collection. And for like optimal results in downstream assays, you really want ideally to process your sample in a single extraction. So we had to be a bit creative to validate the OMNIgene SKIN, and what we ended up doing is we chose to collect paired samples from the left and the right side of the face of healthy donors. and we used like 20 donors per condition. And we use those samples pairs to like assess profile stability and neutrality. And what we were doing is that one of the two samples was extracted at baseline, and that was considered to be the ground truth, while the other one was extracted post-challenge, and challenge would be either simulated shipping or extended storage at room temperature. So just to give you an idea of simulated shipping conditions that we tested, we did 50 degrees for 1 day, 37 degrees for 3 days, or 3 cycles of freeze thaw, and here is a room temperature storage for like 30 days. But what you need to establish first is what is a biological variability, because we cannot expect the left and the right side of the same individual to be exactly identical. So that's what we did, and we measure the profile like differences across our sample pairs. And in that case, biological variability, everything was extracted at baseline. Each dot represents a single donor, and you see that the biological variability is overall quite low. And it's much lower than the donor-to-donor variability, where you're comparing the unique profile of each donor. And when you look at the different conditions that we tested for stability, you clearly see that following simulated shipping conditions or like room temperature storage for up to 30 days, we don't see an increase in the variability, which is like an indication that overall the profile is stable in those donor through those conditions. And I'm still going to show you some 16S taxonomic profile from those very samples to convince you that we have good profile stability. So this is 3 representative donors where I'm showing you baseline versus storage at room temperature for 30 days. And overall, you see that the uniqueness of each donor profile is maintained during storage at room temperature for 30 days. And the samples are not exactly identical, but keep in mind that in our experimental design, we use paired samples left and right. So we knew that the samples were not always going to be fully identical. And this is a taxonomic bar plot showing you relative abundance at baseline or following 37-degree incubation for like 3 days. And you see that there's no significant change in the profile for any of the donors. And we're able to maintain the stability of the profile even when we're incubating at a temperature that could promote bacterial growth. One other important thing that we wanted to measure was we wanted to make sure that OMNIgene SKIN was not going to be introducing any detectable bias in the microbial profile. So we did that using a very similar approach as what we did for stability. And we collected like samples from 20 donors, paired samples, in either OMNIgene SKIN or PBS, and measured like the variability at baseline. And as you can see, when you compare variability in PBS versus OMNIgene SKIN, there's no significant differences, meaning that our collection into our device is not introducing any kind of profile bias. And the variability that we're seeing in both cases is much lower again than the donor-to-donor variability. So we also tested fungal profile neutrality and stability. We chose not to do ITS sequencing because the fungal DNA that you get in a typical skin sample is very, very low. It's only a fraction of the total DNA. So what we ended up doing is using like spike-in of M. globosa, Malassezia globosa, which is like a very well-known skin commensal. And a live culture of M. globosa was spiked into collected paired face samples from 8 donors. And then we measured the amount of M. globosa by qPCR at baseline or following like simulated transport. So freeze thaw 50 degrees or 37, and also room temperature for 30 days. And you see that overall, we don't see any shift in the amount of signal that we're recovering, which is indicating that the fungal profile remains stable over time and during transport. And we also tested neutrality for fungal. So again, we collected in OMNIgene SKIN versus PBS, and you see that we see no differences in the amount of M. globosa we're able to detect, meaning that we're not shifting the fungal profile or the bacterial profile when collecting sample in OMNIgene SKIN. So one of the things that we also wanted to do was to like compare OMNIgene SKIN performance to like other like swab-based skin collection devices. And we did that by collecting a cheek or like forearm sample in OMNIgene SKIN or devices from competitor C or competitor N. We did that for 8 donors. And as you can see, when we collected cheek samples or forearm samples into OMNIgene SKIN, we consistently saw 2 to 3-fold more DNA that was recovered during the extraction than with a competing device. And 2 to 3-fold may not seem like very much, but when you start looking at forearm, which is dry skin, where the biomass is as low as it gets when it comes for skin, a significant proportion of the samples that were collected in competing devices were actually indistinguishable from the environmental control, so uncollected kit. And that's not the case at all for OMNIgene SKIN, where like over 85% of the samples that were collected were at DNA concentration that were well above of the environmental control. And I think that's really like truly indicating that OMNIgene SKIN is optimized for low biomass sampling and other swab-based devices are not. So we also measured like 16S copy number in cheek samples that were recovered when you sampled using OMNIgene SKIN or competitor C or N. And as you can see, we were able to show that we've got improved bacterial DNA recovery. And that's really important because it shows that not only are we getting higher yields, but we're actually getting higher bacterial yields, which mean that there's going to be more bacterial representation in those profiles. And that's better success in any downstream application. So I hope that I've been able to convince you that OMNIgene SKIN is the only validated skin collection device out there. And we've been able to validate its performance for the major type of skin sites, so sebaceous, wet, and dry. We've developed optimized workflow to maximize DNA yields from low biomass sample. And that's really to ensure that there's going to be an increased success in any downstream application. Overall, our kit has a low bioburden, which is good because that means that you can even collect from dry skin where biomass is extremely low, and background DNA contamination is not going to be a big issue. OMNIgene SKIN is able to capture and stabilize the human skin microbiome during transport at ambient temperature and storage for 30 days at room temperature, and that's both bacterial and fungal profile. And importantly, it outperforms all the other swab-based collection devices that we've tested. We're seeing better yields and improved bacterial DNA recovery. And I think the key of improved yields on bacterial DNA recovery is going to give you consistent performance in next generation sequencing pipelines. So we saw a 100% success rate in ITS and 16S amplicon sequencing, but we also were able to isolate enough DNA to enable shotgun sequencing on select higher biomass skin sites, such as like face, scalp, or like toe web, and then you can start taking like a deeper look at the microbiome strains of bacteria, etc. So more out of those samples, really. And now I'll pass on the presentation to Daryl, which is going to talk more about the challenges in skin microbiome analysis.
Well, thank you, Brice. I'm going to talk today about some of the work that we've been doing at Diversigen on the library prep and sequencing portions of the skin microbiome profiling workflow. So as with the human gut, diversity of the skin microbiome is often associated with skin health. And there are also a number of key taxa that can have very large roles and influences on skin health and disease. Brice mentioned one in particular, Cutibacterium, which is often a high frequency or highly abundant component of the skin microbiome in many body sites. So Brice did a really nice job of talking through the optimizations that have been done at DNA Genotek with the OMNIgene SKIN product to optimize sample collection and storage for skin microbiome samples. A lot of the challenges that he highlighted, the fact that skin microbiome tends to be very low biomass and also has often high levels of host DNA present as well. These are also challenges for sequencing the skin microbiome, in particular for doing shotgun sequencing. There are a number of other kind of issues with respect to being able to recover and recover key taxa, such as Cutibacterium, using amplicon approaches that we've done a lot of work on optimizing. So I'm going to tell you about those today. And one of the real advantages that we have in working with DNA Genotek is that we're able to co-develop and co-optimize methods for collection and the downstream processing. And so one of the areas that we've done that is on extraction of the DNA from skin. And so Brice had mentioned a method that was kind of developed internally at DNA Genotek and optimized for extraction of samples from these OMNIgene SKIN kits. We've implemented those methods at Diversigen, and this is some of the data on a variety of different samples collected from different body sites, showing that we can get these robust yields, robust DNA yields from these samples from different body sites. And this is just a summary of data from a variety of different sites where, as Brice had mentioned, the sebaceous or wet tissues that have higher biomass, higher microbial load tend to correlate with better extraction success. But in general, the impact of this co-optimization of both collection and extraction yields to an increased rate of successful collection. And so fewer sample dropouts in your studies and the ability to access, at least for certain tissues, both 16S sequencing and also potentially shotgun sequencing. So there's been a lot of work done in the field on looking at the impact of different variable regions on analyzing the skin microbiome. This is data from a nice paper from Elizabeth Grice's lab from a few years back, where they showed that it's had different performance for analyzing the skin microbiome. In particular, they saw a dropout of one of these critical taxa, Cutibacterium acnes or Propionibacterium acnes, when using V4 primers. They also noted that V1, V3, because of the larger target size, the increased number of polymorphisms within the V1, V3 variable region, did a better job of classifying certain taxa. So in this example, S. epidermidis was able to be classified to the species level with V1, V3, but only classified as Staphylococcus genus using V4. So over the last several years, we've done a lot of work, both at Diversigen and also my work at the University of Minnesota on optimizing skin microbiome, or optimizing 16S amplicon-based microbiome profiling methods. This is data from a paper that we published a few years back, looking at the performance of other methods that are commonly used in the field, like the EMP or HMP methods, for 16S microbiome profiling versus an optimized method that we've developed, where we really tried to optimize the molecular biology to generate more accurate microbiome profiles. And this is data from 20 different organisms. The expected values are shown as dashed line here. And what you can see is that these optimizations that we've introduced really helps to generate a much more accurate microbiome profile. And one of the things that I'll highlight is that we also saw, as with the previous data that I showed from the Grice lab, we also saw a near complete dropout of P. acnes. So this is a log scale. So with some of these existing methods, P. acnes is hundreds of fold underrepresented in this data set. And what you can see is that with some of the optimizations that we've introduced, we're actually able to rescue that dropout of P. acnes. And I'll tell you a little bit more about how that works and how we did that. So in this publication, we laid out a number of best practices for generating amplicon-based microbiome data. These included some kind of straightforward molecular optimizations to improve the quantitative accuracy, such as minimizing PCR cycle number and optimizing DNA template concentration to really try to reduce the effects of PCR biases. Using a highly processive polymerase, which helps to really reduce the amount of PCR chimeras that are formed. And then we also uncovered something that was relatively unexpected, which is the ability to improve also the qualitative accuracy of these methods by minimizing the dropout of taxa. And the way that we did this was by incorporating a proofreading polymerase, which we thought might help to minimize substitution errors and decrease the error rate in sequencing and improve your ability to reliably classify organisms. But it also had the unexpected effect of enabling something that we've dubbed primer editing. And we uncovered this actually in looking at P. acnes or C. acnes in this mock community, where we saw that the reason that this organism tends to drop out in analysis using the V4 variable region is that there's a mismatch between the V4 primer sequences, both the forward and reverse V4 primers, and the C. acnes 16S template sequence. And this mismatch basically completely abolishes the ability to amplify this organism typically. And what we found is that by incorporating this proofreading polymerase, we're actually able to get chewback of these primer sequences and correction of the primers to match the template sequence. And we can see that when we look at the sequencing reads and we look at the region corresponding to the primer sequences, we see an A to G and a T to G change that match the C. acnes template sequence. When we use a proofreading polymerase, but not when we use an enzyme like Taq polymerase that doesn't have this 3' to 5' mix nuclease activity. And so what the impact, the result of this is that we're able to then use primer editing to recover a significant proportion of the expected P. acnes or C. acnes in this mock community sample. So we've seen this with other taxa as well. This is an example from primate microbiome study where there's this particular TM7 taxa that also has a mismatch to the V4 amplification primers that we recover specifically when we incorporate primer editing, incorporate these molecular optimizations into our 16S analysis pipeline. But this taxa is completely drops out when not taking advantage of primer editing. So we've subsequently extended on these observations with respect to the skin microbiome. This is work that's been done at Diversigen by Amanda Sertano, one of our research scientists. And what we found is that for a variety of different body sites, when we incorporate primer editing, incorporate proofreading polymerases into the amplification process, we're able to rescue the detection of C. acnes to levels similar to what's detected with V1, V3. And this is on the right is a plot of paired donors analyzed either with V1, V3 or with V4. These purple bars are the amount of Ketobacterium that are detected in these samples. And what you can see is that we detect very similar levels of Ketobacterium when using V4 as we do with V1, V3, as long as we've implemented these optimizations and enabled primer editing in these samples. So in addition to the ability to detect P. acnes or C. acnes, there are some other trade-offs between using V4 and V1, V3 that are worth considering in designing your microbiome studies. So one of these is the improved taxonomic resolution that you get with V1, V3. And as I mentioned, this is really a result of that increased diversity or increased number of variable bases within the V1, V3 variable regions. It gives you more resolving power. This is visualized really nicely in these plots from this 2019 Nature Communications paper, where you can see that with V4, just because of the smaller target size and the lower entropy of these sequences, typically these can only be classified, most organisms can only be classified down to the genus level. Whereas with V1, V3, you get a lot more organisms that are able to be classified to the species level. The trade-offs, though, are that there are some technical challenges in sequencing V1, V3. So this amplicon is really at the edge of what can possibly be analyzed using Illumina sequencing technology. And so you tend to have some impacts on sequence quality and the ability to effectively stitch those reads together. Whereas with V4, because it's a shorter amplicon, you're able to basically sequence the entire amplicon in both directions, and that gives you improved sequence quality. We've also found that longer amplicons tend to have systematically worse sequencing quality across a range of cluster densities. So for the sequencing aficionados out there, you may know that as you increase cluster density, increase yield, you tend to see lower quality scores. And you can see that trend in these plots. But what we found is that across a range of cluster densities, for V1, V3 tends to have lower sequencing quality than V4. And this can impact both your ability to stitch those reads together and the number of reads that you can effectively analyze in those samples. And then ultimately your mapping and the reliability of your ability to map these reads to a database. So to sum up, today Brice and I have told you about work that we've been doing across this workflow to optimize the collection of skin samples and storage and stabilization of those samples. Work that we've done to optimize extraction yields and optimize the proportion of these samples that can be analyzed. And then I've told you about the work that we've done to optimize 16S profiling methods, in particular incorporation primer editing, which allows you to expand the number of taxa that you can detect with a given primer set. And for skin, this is really critical, at least when using V4, because it allows you to detect Ketobacterium acnes, which normally would drop out of this analysis. And these improved extraction yields allow us to not only to access a lot of these samples for 16S or amplicon sequencing, but also open up the possibility of doing shotgun sequencing on a large number of samples. And so at Diversigen, we're currently offering both V4 and V1, V3 analysis for skin microbiome samples. And we have a Data 2 based ASV analysis pipeline for doing the actual classification and analysis of that data. So with that, I think Brice and I are happy to take any questions you might have. Thank you.
Perfect. So we've already received some questions through. And I think the first, Brice, is probably best directed at you. So actually, we had a few questions here, which all concerned other uses for OMNIgene SKIN, so either other omics or other sample types, so non-human or even viral work. So don't know if you want to address those in a bundle.
Yeah, I probably can. So first question that we have is a stabilization buffer in OMNIgene SKIN compatible with other omic analysis, so proteomics, transcriptomics? So the chemistry that's in our tube is compatible with RNA, and we've tried looking at RNA from skin bacteria. And to make a long story short, I believe that we were able to get some RNA out of the highest biomass skin sites, and the quality was pretty good, but I do believe that biomass being so low on skin, it's very hard to get enough RNA for transcriptomics in general. But if someone was to look at the expression of like a given transcript and use a qPCR assay, I think that would be something that would be completely feasible. So a targeted approach instead of doing like full RNA-seq. And as far as proteomics, I think it's something we've never looked at. Our chemistry tend to be lytic, so I would say that protein would not remain intact, but we haven't really looked at that. And the second question, so did we consider like sampling like animals, so pets? I think that's not something we've done, but based on the data that we got from the scalp, I would expect that the performance is going to be about the same as what we saw with a human scalp. And like we would be actually really interested in finding out if anyone tries.
Perfect, thank you. And we actually had two questions here regarding the wetting of the swab prior to collection and different potential compositions of wetting buffers. So I don't know if you want to comment on our experience with wetting swabs and wetting buffers.
So yes, so the first question is about using a low concentration of a detergent. So this is something that was part of the original HMP protocol. So SCF1 does contain like 20. We've compared the performance of SCF1 to the weighting agent we're using in the final form factor in our kit, and we didn't see much of a difference. So we're currently using a detergent in the weighting agent, but we've seen similar performance. So we felt that it was not a requirement. And the second question is a wet swabbing the key to increase yield versus competitors? So the answer is going to be no. For the competitor collection, we ended up following the HMP collection protocol for skin. So we were actually using SCF1 for those collections, and the swab was pre-wetted, same as OMNIgene SKIN. So we believe that the real difference in terms of yield versus our competitors is all the optimization that we made around sample processing and extraction. So making sure that we get good release from the swab into the extraction, but then also using optimized workflows so that you get as much DNA as possible from every sample.
Thank you. I see we actually have two regarding the primers, specifically V3, V4. So maybe this is better for you, Daryl. So we have one asking whether we tested primers targeting V3, V4 commonly used for human microbiome, and the reason why we don't use V3, V4 for 16S sequencing.
Yeah, so I mean, there's quite a menu of different variable regions out there. You know, different fields, different groups have settled on different variable regions for different reasons. I mean, with skin, there's definitely some advantages, to using certain variable regions for resolving, certain taxa that are common on the skin microbiome. So we have, or I in the past have experience running a whole bunch of different variable regions. In academic work that we've done at the University of Minnesota. We're also in the process of kind of doing a very systematic sweep of all the different regions that are commonly used in the literature against a variety of different sample types. And so while that's something that we're not currently offering at Diversigen, I think that in the future, we might well be kind of expanding that menu of variable regions. In general, all of these variable regions are going to have, kind of similar trade-offs and limitations, where they'll be good for resolving some taxa and not for others. And really, in order to get down to that, kind of species or strain level resolution, one often needs to be able to do that reliably, one needs to kind of take a shotgun sequencing approach.
Perfect, thank you. I see we have another one, Brice, that might be for you, regarding the bioburden that is present in the collection system. So the question is asking, it looks like some sites have the same realm as the DNA concentration, and how we can comment on the bioburden present in the kit, and if we know where it's coming from.
Yeah, I think that's an excellent question, and that's really an issue that's going to be popping up for anyone that's doing low biomass sampling. And when it comes to skin, when you're looking at dry skin, so forearm or legs, for example, we're only talking about a few thousand bacteria per square centimeter. And if you make the conversion in the sheer amount of DNA you can extract from such a sample, that would be like a few nanograms. So bioburden is definitely going to be an issue. And for samples collected from the farm, even in OMNIgene SKIN, the yields were extremely low. So we're talking about 10 to 20 nanogram per kit for the entire sample site. So that's very low. At the same time, for like over 80% of the samples, those DNA quants were higher than the environmental controls. And we did the testing downstream for amplification with ITS and 16S. We did see big differences in the final yield. So that means that the bioburden is not always DNA that's going to be amplifiable by PCR, which is good when you're using targeted sequencing, 16S and ITS in that case. In terms of where that bioburden is going to be coming, so we're asking the donor to collect from his skin inside his home for like a minute. There's the DNA that's coming from that step, so we have a breaking point, but donors could be like touching the swab below the breaking point and end up contaminating. You can touch surfaces. In general, if you do a good job during the collection, there's going to be very, very low amount of contaminating DNA. And that's good because that means that, as I was saying, bioburden is overall low, but not completely absent in our kits. And another source of bioburden, which is significant, and there's no way around it, is the extraction kit. So you're going to have to be extracting the DNA from the samples. And every extraction kit, there's a few papers showing that there's a ketone, which is basically the DNA coming from the extraction kit. And I think, again, like the only point I can stress is that if somebody is interested in doing skin sampling and so on, you just have to make sure that the sample is collected as well as possible so that you're going to have as much DNA collected from that site as possible so that your downstream analysis is successful, really.
Great, thanks. And I think this might be a one for both of you, since both of you took part in validation and in analysis that happens at Diversigen. So we had a question that was submitted prior to the webinar regarding how to increase the microbiome signal versus the human signal. And then we had a question submitted during as well regarding whether the human sequences were removed for analysis. So I don't know if how you guys want to tackle that one.
I think Daryl is probably a better place to answer those than I would be.
Yeah, so in general, you know, we do have a host removal step in our analysis pipeline. And so that's one of the things that you contend with, you know, doing shotgun sequencing in the skin is that there can be a large amount of host material. You know, I know there's been work at DNA Genotek on, you know, host depletion methods, which, you know, have some trade-offs and have, you know, mixed, you know, success in being able to actually deplete hosts in sort of an unbiased way that's not changing the profile of your sample. And so in general, you know, a good solution is just to basically use brute force and you know, sequence these samples a little more deeply. One of the things that Helly's mentioned in the introduction was the shallow shotgun approach that we've pioneered initially at CoreBiome and now at Diversigen. And that approach really has some nice advantages when it comes to doing shotgun sequencing in samples that have a lot of host contamination or have you know, low biomass, or the you know, where you don't have the ability to get a huge number of reads that are mapping to bacteria, because that data effectively always is then shallow data when with respect to the microbiome component of the sample. And so some of the optimized pipelines, the optimized mapping workflows that we used for that shallow shotgun data can be really useful for analyzing skin samples. So where you basically the process is to initially do a host removal step to get rid of the human reads, and then to use these optimized alignment methods to get as much signal as you can from that shotgun data.
I did see we had some questions also regarding, you know, our experience with very specific diseases and health statuses or special use cases. And I'm just going to jump in as product manager here and comment that obviously we haven't tested across the board for all of the different potential health statuses and diseases out there. We have some studies on the go, but anybody who has a specific study that they want to run because that's your research interest, we are more than happy to provide you some sample kits so that you can start testing this new method and see if it works specifically for you. And we will help you troubleshoot from collection to analysis, since we have the expertise in-house clearly as presented during this webinar. So just wanted to throw that out there. And I think that's it for the live questions. I'm just going to give one more check to see what we have. And we did get asked if we would share the slides, and don't worry, everybody who is still here, we will be sharing the slides at some point, so you can see those. And I believe we'll be diffusing the presentation at some point as well for everyone to watch again. I'll just give it another minute in case there are any additional questions that come through. And we want a big thank you to everyone for taking part today. Thank you to Daryl and Brice for presenting. This was great. And I appreciate everyone's time. So nothing else has come through so far. If anybody does have additional questions, you can definitely reach out to DNA Genotek or Diversigen. We've got lots of different options on our websites for you to get in touch, and we'll be happy to answer your questions that way. And we've also displayed a link so that if you're interested in trying the OMNIgene SKIN kit that we've just mentioned today, you can click on there, or again, just visit the DNA Genotek website, and we'll put you in touch with the right people to hand you those. And with that, thank you everyone.
Thank you.
Thanks.